First of all, thank you very much for staying till the last talk. Thank you very much, especially to Eddie de Robertis for this invitation, and to Monsignor Marcelo Orondo, thank you very much. So during the past 5 years, we have witnessed a revolution in cancer immunotherapy. Since the first observations by William Coley that it is possible to stimulate the immune response against cancer, it took almost 120 years until we fully realized and fully appreciated the power of cancer immunotherapy and the concept of cancer immune surveillance. So how does it work? Tumor cells release tumor antigens. These tumor antigens are taken up by antigen-presenting cells. These antigen-presenting cells, mainly dendritic cells, present these antigens to naive T cells. These naive T cells get activated, proliferate, and migrate to the tumor to kill the tumor at the tumor site. But during the course of the disease, these T cells get dysfunctional and also exhausted, because several immune escape mechanisms and immune inhibitory proteins that can block them and also induce apoptosis. So there are several pathways and several mechanisms, several inhibitory pathways that control these effects. They are called inhibitory checkpoints. So inhibitory checkpoints contribute to silence Th1, Th17, and CD8 positive T cells, which are the most important cells to kill the tumor, but these are also important cells that cause autoimmune diseases. Also, immune checkpoints contribute to expand and recruit regulatory T cells and also to convert immunogenic into tolerogenic dendritic cells. So blocking, using specific antibodies, blocking immune checkpoints, including PD-1 or CTLA-4, increases the immune response both in experimental models, but also in patients, increasing survival, overall survival, and also progression-free survival in many types of patients. So this is a comparison of patients treated with chemotherapy versus patients treated with nivolumab or pembrolizumab, which is the anti-PD-1 antibody. So blockade of inhibitory checkpoints increases the immune response against tumors. So most of this information has been reached by analyzing the genome, proteome, and also the transcriptome of the tumor microenvironment, but we know much less on how the glycome, which is the collection of sugar structures in cells and tissues, contribute to the tumor microenvironment. And this can be related to the complexity of the glycome, but far from the traditional idea that glycans decorate the surface of all mammalian cells, the glycome has the potential to display 100 million more information than the genome. And this is even more complex in the tumor microenvironment. So probably the main goal of our group is represented here in this review we wrote for immunity, is to decipher the glycocodes in immunity. So the responsibility of deciphering this biological information encoded by the glycome is assigned to endogenous glycan-binding proteins, including galactans. There are C-type lectins, C-glecs, but I will speak today about galactans. Galactans are a family of soluble glycan-binding proteins that have affinity for multiple N-acetylgalactosamine, which is galactose beta-1,4 and acetylglucosamine units present in complex in glycans and also in core glycans. They are highly conserved throughout the animal evolution, from fungi to mammals. They are secreted through a non-classical and conventional pathway, ER-Golgi independent pathway, very similar to IL-1 beta or FGF. And they act through binding to different glycosylated receptors and forming lattices that control segregation trafficking of these glycosylated receptors. They are classified into 3 different families: prototype galactans, tandem repeat galactans, and chimera-type galactans. We will focus today on prototype galactans, particularly on galactin-1. So using a polyclonal antibody that reacted against chicken glycoconjugates in the retina, we found an unexpected finding in 1993. We found a protein band of 14-KDA that was regulated in macrophages in antigen-presenting cells, and now we know it's galectin-1. We sequenced, we cloned the gene, and now we know it's galectin-1. It's an oncovalent homodimer composed of subunits of 14-KDA. It is expressed in antigen-presenting cells, including macrophages and dendritic cells, promotes T-cell exhaustion and apoptosis, and its expression is markedly upregulated in the tumor microenvironment and correlates with the acquisition of metastatic phenotypes. So we thought that probably tumors may use galectin-1 to evade immune response. So in this paper we published in 2004, we found that melanoma cells increased the amounts of galectin-1 to selectively eliminate activated but not resting T cells. So when we eliminated galectin-1 from the tumors, there was a reduction of tumor growth, and this effect was prevented when we inoculated these mice with anti-CD4 or anti-CD8 antibodies. So there was a dialogue between galectin-1, CD8, CD4 T cells, and the tumor. We call this the sweet kiss of death. So we confirmed this finding in Hodgkin lymphoma. Hodgkin lymphoma is composed by Reese-Steinberg cells. Reese-Steinberg cells are surrounded by non-productive infiltrates, and we found that, that Reese-Steinberg cells produce very high amounts of galectin-1 that contributed to create a Th2-skewed microenvironment. It was expressed in classical Hodgkin lymphoma, but not in other types of lymphoma. And when we analyzed the mechanisms that lead to regulation of galectin-1 expression, we found that an AP1-dependent enhancer contributed to regulate galectin-1 expression in Reese-Steinberg cells. Most recently, there were many papers from different labs demonstrating that galectin-1 can activate immune evasive programs in different tumors. So galectin-1 is not only produced by melanoma cells and Hodgkin lymphomas, but also in many other different tumor types, including glioblastoma, T-cell lymphoma, lung adenocarcinoma, pancreatic adenocarcinoma, and others. So we wanted to explore the mechanisms by which galectin-1 can bind and cause exhaustion and dysfunction of T cells. So Marta Toscano from my lab, she decided to explore the mechanisms that galectin-1 was functioning. So galectin-1 binds to complex N glycans, to lactosamine structures in complex branch N glycans, and also in core-2O glycans. Here you can see that galectin-1 likes yellow and blue, so those who know Argentina know that the best colors in Argentina are yellow and blue. Those who like soccer? No. Okay. So what Marta found was that during differentiation, Th1 and Th17 cells that are important for the anti-tumor response, they express all the repertoire of glycans that are important for galectin-1 binding, so core-2O glycans and branch N glycans. In contrast, Th2 cells and also naive T cells express very high amounts of alpha-2,6 sialic acid, which prevents galectin-1 binding. At the same time that Marta was exploring T cells, Juan Martini Larregui was exploring dendritic cells. And he found that galectin-1 bound to dendritic cells and converted immunogenic dendritic cells into tolerogenic dendritic cells that are unable to present antigens to T cells. So he defined molecular mechanisms in which galectin-1 binds to CD43 on dendritic cells. These dendritic cells produce very high amounts of IL-27, and IL-27 converts effector T cells into regulatory T cells. So by any way, galectin-1 is a stop signal, stops T cells and stops dendritic cells. So Mariana Salatino and Tomás Dalotto, also from our lab, they demonstrated that in the case of breast tumors, galectin-1 contributes to metastasis by increasing the expansion of regulatory T cells. And recently, Santiago Méndez-Cuerco tried to dissect the glycosylation signature of regulatory T cells. And regulatory T cells, they have very low amounts of tetraantenary N glycans and very high amounts of alpha-2,6 sialic acid. So galectin-1 binds to cytotoxic T cells, to Th1 and Th17, but not to regulatory T cells. So it kills selectively the anti-tumor T cells. So there is a long-standing question in immunology. Why doesn't your mother reject you? So our galectin-1, we suspected that maybe galectin-1 may also contribute to immune tolerance and prevent maternal immune attack of the growing fetus, because the growing fetus in pregnancy is very similar to tumor immune escape. And we defined an immunosuppressive cascade that is initiated by progesterone in the trophoblasts that contributes to immunosuppression, and galectin-1 knockout mice had increased amounts of fetal rejections, and also women with spontaneous abortions, they have higher amounts of anti-galectin antibodies and also low amounts of soluble antibodies, or soluble galectin-1. So when we blocked galectin-1 expression in the tumors, all the pathologists told us there is an increased immune response, but also there is a reduction of blood vessels. So you should look at angiogenesis. So then we started to study a little bit of angiogenesis, and we found that immunosuppressive and pro-angiogenic programs were integrated and co-regulated in cancer and inflammation. So some pro-angiogenic factors are also immunosuppressive, and some immunosuppressive factors are also pro-angiogenic. So Diego Crossi decided to explore the role of galectin-1 in endothelial cells during immune vascular programs. And so he first defined the glycosylation signature of endothelial cells. He found that galectin-1 bound to endothelial cells in a dose-dependent manner. And when we use a galectin-1 that lacked carbohydrate binding domain, we lacked binding of galectin-1. Also, galectin-1, we found a partial prevention when we silence GNT5 or C2GNT1. So complex and glycans are important for this binding. We found that galectin-1 induced angiogenesis to similar levels of BGF, which is a canonical ligand that induces angiogenesis of endothelial cells. And this effect was blocked when we blocked the glycosyltransferase GNT5. So looking for a potential receptor on endothelial cells, we found, surprisingly, that galectin-1 selectively bound to BGF receptor 2, which is the same receptor that shares with BGF, with vascular endothelial growth factors, and trigger phosphorylation of BGF receptor 2, and also dimerization of the receptor. So when we block different receptors, including BGF receptor 1, 2, 3, and also integrins, only blockade of BGF receptor 2 prevented galectin-1 binding. So when we perform site-directed mutagenesis of the receptor, the BGF receptor is composed by 7 domains and 18 N-glycosylation sites. So we decided to generate mutants of each of the 18 N-glycosylation sites, and we found that galectin-1 bound to receptor to domain 3, 4, and 7, but BGF binds to domain 2 through protein-protein interactions. So thus we wonder, we concluded that galectin-1 was overexpressed in tumor cells in response to hypoxia through mechanisms that were independent of HIF and independent of BGF. It bound to BGF receptor 2 and triggered BGF-like signaling, mimicked BGF-like signaling. So we wonder whether galectin-1 glycan interactions may promote BGF-like signaling in anti-BGF resistant tumors. And this is a very clinically relevant problem because most patients develop resistance to anti-BGF, which is bevacizumab Avastin therapy. So we first inoculated tumors that are resistant to anti-BGF treatment into mice, lung tumors, T-cell lymphoma, the LLC1, the R1.1. We measured galectin-1 expression, and galectin-1 was dramatically upregulated during the day 4, which is the day of hypoxia after anti-BGF treatment, in lung tumors, in T-cell lymphoma, and also in pancreatic genetically engineered model. And it was very specific because there was no increase during melanoma injection, which is a tumor that is sensitive to anti-BGF treatment, and also there was no increase of galectin-3 and galectin-8. So we wanted to explore the glycosylation of endothelial cells in vivo. So we inoculated mice with anti-VEGF. We isolated by sorting endothelial cells surrounding these tumor cells, and we analyzed the glycosylation signature of these endothelial cells. So as you can see here, there was a decrease of alpha-2,6 sialic acid in those tumors that were resistant to anti-VEGF and were treated with anti-VEGF antibody. So galectin-1 can bind to these endothelial cells. So not only there is an increase of galectin-1 expression, but also the windows are open for galectin-1. So we decided to explore whether galectin-1 glycan interactions may play a role in resistance to anti-VEGF. We inoculated tumors that are resistant to anti-VEGF, lacking galectin-1, and as you can see here, those tumors that lack galectin-1 expression converted tumors that were refractory into tumors that are sensitive to anti-VEGF treatment. And this effect was even increased in galectin-1 knockout mice. So we wanted to see the same effect, but on the other side of the street. We inoculated tumors that were resistant to anti-VEGF, but in mice lacking N glycans in endothelial cells. And the same effect was observed. Tumors that were resistant to anti-VEGF were now sensitive to anti-VEGF. So we asked the opposite effect. What will happen if we inoculate tumors that are sensitive to anti-VEGF treatment in tumors in mice lacking alpha-2,6 sialic acid, in mice lacking the ST6-Gal1 allele transferase? And in this case, tumors that were sensitive to anti-VEGF, they are now resistant to anti-VEGF treatment. So in summary, in those tumors that are sensitive to anti-VEGF treatment, endothelial cells are covered by alpha-2,6 sialic acid, and galectin-1 cannot bind. In those tumors that are resistant to anti-VEGF, so these endothelial cells express very high amounts of beta-1,6 and glycan branching, low amounts of alpha-2,6 sialic acid, and galectin-1 can bind to immunoglobulin domain 3, 4, and 7, and trigger VEGF-like signaling and promote angiogenesis. So these studies were published by Diego Crossi and Juan Pablo Serliani in 2 different papers in 2012 and 2014. So very recently, we asked about whether galectin could be a good biomarker of anti-VEGF therapy, and we used plasma from patients that were exposed to the AVAST-M, which is the large melanoma trial evaluating angiogenesis inhibition. So to validate this, we took 189 patients, 94 patients on the bevacizumab arm and 95 patients on the observation arm. First, we found that there was no significant interaction with the trial arm in predicting overall survival, so there was no intrinsic resistance. But when we did the longitudinal GAL1 plasma analysis, we found that galectin-1 increases, the levels of galectin-1 over time are associated with an increase in recurrence and the increase of the risk of death in the bevacizumab arm. So we also found that galectin-1 is also part of a regulatory circuit that is stimulated by flagellated microbiota, and galectin-1 increases in myeloid-derived suppressor cells and contributes to immunosuppression in these tumors, and all this circuit is regulated by the SATB1 transcription factor. So we wondered whether we could capitalize galectin-1 for cancer therapy. So we designed first glycoamines that blocked all members of the galectin family, then small molecule inhibitors, and finally anti-galectin-1 neutralizing antibodies. So the anti-galectin-1 neutralizing antibodies block angiogenesis, block phosphorylation of VEGF receptor 2, combination of anti-VEGF plus anti-galectin-1 suppressed and converted refractory into sensitive tumors, but probably more importantly, increase the amounts of the magnitude of the immune response, particularly Th1 and Th17 response, and also CD8 positive response. So also when we blocked MGAT5, so also we found an immunostimulatory effect was recapitulated what found with galectin-1 blockade. So we also found that because of the increased immune response in the tumors, we wonder whether anti-galectin-1 treatment may also increase the anti-PD-1 treatment. And as you can see here, synergistic blockade of anti-PD-1 and anti-galectin-1 has a dramatic effect on tumor growth in those tumors that were resistant to anti-PD-1. So galectin-1 acts as an immune inhibitory checkpoint that promotes dysfunction of Th1, Th17, and CD8 positive T cells, convert regulatory T cells into tolerogenic dendritic cells, also promotes regulatory, expands regulatory T cells, increases links tumor hypoxia and angiogenesis, and also promotes M1 macrophages, increases to polarization to M2 macrophages. So to define an immune checkpoint, it's important also to define whether it can be therapeutically beneficial for the treatment of autoimmune diseases. And very early, we found that administration of galectin-1 or gene therapy using galectin-1 suppressed disease in several models of autoimmune inflammation, including arthritis, experimental autoimmune encephalomyelitis, uveitis, colitis, hepatitis, and diabetes. We found that galectin-1 knockout mice have enhanced susceptibility to these autoimmune diseases, and more recently, Marta Toscano and Veronica Martinez Aló found increased spontaneous autoimmunity in aged galectin-1 knockout mice. Also, Sarah Staronson found that galectin-1 in the central nervous system was produced in astrocytes during the resolution of inflammation in a multiple sclerosis model. Galectin-1 bound to CD45 on microglia cells and converted microglia cells to M2 microglia cells, which prevented neurodegeneration. So we ask, if we block galectin-1 expression, then we can increase the immune response against tumors. But then, if we treat mice with galectin-1, we can suppress disease. But the problem of inoculating galectin-1 is that galectin-1 is very unstable, and it's degraded very easily. So we received the help of many chemicals that came to our lab, Santiago Di Lela, particularly Charlie Guardia, and Alejandro Cagnoni. And we decided to explore by molecular modeling what was the problem. And the problem why it's unstable and we need very high concentrations to suppress autoimmune inflammation is that high concentrations are needed to bias the equilibrium toward the dimeric form. Also with the help of Rafael Radi, also with Dario Estrin and Julio Caramello, we found that galectin-1 is very sensitive to oxidative inactivation and also very sensitive to acidic pH. And we observed that it's a very small protein, but there are 6 cysteine residues in these proteins, and 3 are very important and confer sensitivity to oxidation and also sensitivity to acidic pH because 1 histidine residue in the molecule. So we decided to generate different mutants, and with the different mutants, we selected 3 different mutants that at very low concentrations, they were very resistant to oxidation and also to acidic pH. We call them the super puff girls, the super galectin-1 proteins. So as you can see here, this is a model of arthritis, this is a model of multiple sclerosis, the EIE model, and this is a model of inflammatory bowel disease. So the super GAL1 protein had a dramatic inhibitory effect on collagen-induced arthritis on the EIE model, and also a recovery of weight in the inflammatory bowel disease. And what's very interesting was that each super GAL1 protein has a particular specific effect. And so we found that by generated mutations, the glycan binding specificity changed. So to define an immune checkpoint, it's also very important to study these checkpoints during infection and during allergic inflammation. And very recently in our lab, Carolina Poncini defined that Trypanosoma cruzi uses galectin-1 to evade the immune response, and Roberto Davicino found that Yersinia enterocolitica and enteropathogenic bacteria also co-opts the galectin-1 inhibitory pathway. And also in collaboration, we recently showed that galectin-1 contributes to suppression of allergic airway inflammation. So galectin-1 behaves like Dr. Jekyll and Mr. Hyde. So we call this the suitcase of Dr. Jekyll and Mr. Hyde. The suitcase because it binds beta-galactosides in some cells. So galectin-1 does always the same, creates tolerogenic dendritic cells, promotes dysfunction and apoptosis of Th1, Th17, and CD8 positive T cells, converts regulatory T cells, also polarizes macrophages toward an M2 phenotype, and also links hypoxia to angiogenesis. But during cancer and also infection, it behaves as Mr. Hyde. And during autoimmune disease, it's Dr. Jekyll. It's the good guy. So this is the most important slide. This is my group in IBIME, Instituto de Biología y Medicina Experimental in Buenos Aires, and the funding agencies and all our collaborators. Thank you very much.